Good afternoon, everyone. And welcome to our webinar today. My name is John Waterman-Smith. I'm the VP of Business Development. Briefly, I would like to introduce you to the speakers and panelists for today's webinar. Today, we're very fortunate to have Professor Rob Kagan and Professor Marshall Posner presenting how the personal discovery process was developed and how it's being used today to help patients. Laura Towett, our CEO, will then explain how to access the personal discovery process for your patients. Professor Ross Kagan is Regius Professor of Precision Medicine at the University of Glasgow and was previously at Mount Sinai and the developer of the personal discovery process. We have Professor Marshall Posner. Marshall is Professor of Medicine and Medical Director of Head and Neck Oncology at the Icahn School of Medicine at Mount Sinai. Laura is our Founder and Chief Executive Officer, and we also have Nal Villegas, who's our Chief Scientific Officer. We also have the pleasure of Josh Chow and Stephen Bernardi, who are joining us from Medicision. And Medicision are our partner in Australasia. And now Josh, Stephen will be our panelists, along with Ross, Marshall, and Laura at the end of the webinar to answer your questions. And now, without further ado, I'd like to pass the baton over to Professor Ross Kagan to begin our webinar today.
So first of all, John, thank you very much. This is a particular pleasure for me. This is something I'm very passionate about. And so this is very helpful. And thank everybody for attending this Zoom meeting today. This seems to be the new format that we are getting used to. And of course, we are talking today because of the challenges we have with cancer. So there has been enormous cancer advances. I think we can all agree to that. Cancer death rates have been coming down over the past probably 30 years. And honestly, most of that until recently was due to a decrease in smoking and increase in colonoscopies. There has been some recent impact by some therapies, but still cancer mortality rates really have been moving very slowly. And it is now joined cardiovascular as a major killer in the developed world. And it's very frustrating to put a point on this. If you have a tumor, solid tumor is the biggest problem, and you fail chemo and radiation, there aren't a lot of good options for about 2/3 of patients. So 2/3 don't really respond to current targeted therapies. And what I'm going to do over the next several minutes is I'll talk about some of the problems that my colleagues and I have found in trying to understand why therapies have had trouble moving from bench to bedside, and we'll talk about a solution. Next slide, please, John. Great. And the key point here, and it's said very nicely here, cancer is in fact a complex disease. And as we've struggled with therapeutics, I think everybody has gone back to the drawing board and sort of rediscovered this fact, which has been known for quite some time, but has not maybe been fully appreciated its impact on cancer therapeutics. Cancer is a whole body problem. You're growing a part of yourself, and attacking that is complex, and it has very complicated interactions with the rest of the body. And also, and importantly, cancer is not driven by a gene or a protein. We often talk about the driver of the tumor, but in fact, tumors, like everything else in your body, are driven by gene networks and by networks in general, body networks and so on. And I think attacking cancer as a problem of a protein really has struggled, and that is because cancer is complicated and it's really a problem of networks. So in the next slide, this point has been made many times. Many of you probably have seen many lectures talk about the struggles of precision medicine. Precision medicine defined by sequencing a tumor, identifying a druggable target, and then taking a clean drug and hitting that target. That has worked overall about 4 to 5% of patients. And that number actually has not changed very much over the past few years. It's really been a challenge. Many colleagues, and especially, and certainly my laboratory, we spent a lot of time looking into this, and what we've concluded, which I won't be able to go into in detail here, mercifully, is that it's really the complexity of the genomics of the tumor that is a key problem in its treatment. A lot of genes drive tumors. Targeting one gene is going to struggle against that sort of network onslaught. And so that takes us to cut to the chase, which is once we became convinced that a key problem in drugging tumors is that they're complicated, which seems obvious, we decided to try to do something about that. And that's the personal discovery process you're going to see here that Laura and her company has really developed, building on what we've done. They've really built beautifully. So if you can go to the next slide, please. This is an overview of it. I just want to point out some of the key steps here, which is that this technology is really the result of a decade of work focused on just building the technology behind this. We've been awarded significant funds from the NIH and from other grants. We've been very fortunate in that. And also, I'm genuinely very excited to partner with Laura, somebody I admire quite a bit, in bringing this technology much more broadly. It's actually, as opposed to the small effort that we've done in Mount Sinai. Next slide, please. This is the scientific team. We'll get to the other scientific team in the next slide, which is the fly. And I just want to make a quick point here that this would not be possible without a large team of people. We recently published a paper that, a patient report that had 29 authors on it. So this is really a team effort. So a network approach deserves a network of people working on it. And in addition to myself and my laboratory, Eric Schott has played a key role. He's one of the top genomics persons on the planet, and he has been instrumental in converting genomic reads into what we can use to create our flies. Marshall, Chris, Selena, these are oncologists that have translated our bench work into something that's actually useful and can treat patients in a safe and effective manner. Next slide, please. And this is a summary of the process itself. So it's a bit complicated, but actually at its core, it's pretty straightforward. When a patient comes to the clinic and they are diagnosed with a particular cancer, let's say, for example, colorectal cancer, we've done quite a number of different types of cancers. If a patient is diagnosed and they choose to step into the PDP process, we begin with a really full and sophisticated genomic analysis. This is really a little more sophisticated than I think most people are used to. And from that, we actually create a tree of likely important genes in the cancer, not just cancer drivers, but other genes as well that are likely to affect drug response. Once we have that set, typically 5 to 15 genes that we're going to need to alter in the fly to model that particular patient, then each patient gets its own personalized fly avatar. And for example, if the patient has colorectal cancer, we will alter 10, 12, 15 genes in the fly gut, the same genes or the analogous genes to what we see in the patient. Once we do that, the fly actually gets aggressive metastatic colorectal cancer in this case. And my laboratory has published some significant publications showing this and exploring that. But once we establish that fly, now we have a magic tool. We actually grow often hundreds of thousands of these flies for each patient. This is why flies are really the only animal system you can do this. And we do that because we are screening through multiple rounds, really most of the FDA-approved drugs available. That's cancer drugs, antidepressants, anti-arthritis drugs, and so on. All of them are screened through multiple rounds to create typically a 2 to 3 drug cocktail. That is our network approach to the tumor. This cocktail is usually a novel cocktail, but of drugs shown to be safe. It can often be a combination of cancer and non-cancer drugs, so they'll be unique, and they'll be unique to that patient. And once we identify that cocktail and we vet it through tumor boards to make sure that it is safe for that patient, we then offer that treatment as an option to the patient, and they can choose to take it. Next slide, please. So all of this together is to say that what we're doing is taking a network approach to a complicated network problem, cancer. And I like the image here of a network because it really sort of hammers home the fact that we are not just picking one target or even just picking 2 or 3 targets. We're using a cocktail of drugs to move a disease network back towards a healthy network. And this is a very different approach than precision, which is more stabbing the network. We're moving that network And through this method, we think also it can be less toxic because you're working within the confines of the body. You're respecting its network while going after the tumor's problems. Next slide, please. Thank you. And so just a couple more slides. For those of you who are pondering why flies, I see it says here, why flies? And there are many reasons, and this would be a long discussion. I love flies. I've been a fly boy my whole life, but not just because I love them. Actually, many, probably the majority of genes that drive cancer have fly names were discovered in Drosophila. Many of you may remember it from high school or college, crossing them together. It's really a classic system that thousands of researchers use. But most importantly for what we're doing, they're really the only system to get to this level of network sophistication. The idea of making a 10 or 15 hit model in a mouse or a zebrafish and then growing up 400,000 of them, that's just really not going to be possible. In flies, we can make this possible because of the advantages that a century of researchers and tool building have done to really make the fly useful. And the image on the right just notes that most of the major organs that we have, flies have. Last slide, please, for me. And so I'll finish by saying this isn't just pure theory, although we're definitely running out on the edge. We do, and others, but especially we do have some success using flies to tackle difficult problems. My laboratory first got into cancer and into this problem looking at a thyroid cancer it's actually can be a deadly one, multiple endocrine neoplasia type 2, and medullary thyroid carcinoma that derives from it. And long story short, we modeled that in flies. We used that to screen candidate drugs, and building on others' work, such as Massimo Santoro's work, we helped identify vandetanib, now has a name, vandetanib, as a useful drug for treating this. That went into clinical trials. The people who put it in clinical trials were actually on our original paper, Sam Wells and AstraZeneca, and it was approved in 2012 as the first and still one of the standards of care for medullary thyroid carcinoma. So flies can be useful, and what Laura and her company are doing are really taking this technology and pushing it much further to try to embrace more of the complexity of cancer. So thank you, John.
Thank you, Ross. We'll move on now to Marshall.
Thank you. I'd like to expand Dr. Kagan, Ross's presentation. When he first came and presented this concept to me many years ago, I thought it was absolutely genius. One of the biggest problems in development of drug therapy for patients is having an economically feasible system, and being able to do it for individuals is almost beyond the economic abilities and resources of any particular institution. So I was very pleased to be a part of this and to be a part of a team and to work with his laboratory to help develop this program. And now to have it operationalized, I'd like to really thank Laura and her team at My Personal Therapeutics. Operationalizing this, I think, is going to provide new information and actually therapeutics for individuals and a larger population of patients as more data is obtained. So when we began, we really went where no men had gone before, which is to really develop a program to do N of 1 clinical trials where we would develop an individualized therapy. We were not attempting to change the indications for any particular drug, so the FDA really allowed us to put together drugs that had been in people, so we have used primarily approved drugs. And through our screening process and our research development process, we elected to focus on colorectal carcinoma as a relatively large patient population with a fairly long survival, giving us the opportunity to really develop the personalized fly. As we evolved our program, we decided to really focus on KRAS mutant patients, as this would provide us with a group of patients that would really benefit if and when we developed a personalized therapy for each of those people and individuals. Colorectal carcinoma, KRAS positive colorectal carcinoma is extremely difficult to treat. Very few drug combinations work in this, and we see patients literally progressing within 9 months to a year of the development of a particular protocol or treatment for them, and then not responding to further therapy. So it's very typical of what Ross talked about in terms of the resistance of tumors to single agent or even to multiple combinations, as they are chosen primarily based on historical results. So the average, you can see here some data on. You can see here some data on the number of patients with colorectal carcinoma who have KRAS mutations. About 2/3 of our Sinai patients, as a referral center, were KRAS positive, and so we identified this as a need. In addition to the KRAS mutations and other shared mutations that are part of the development of colorectal carcinoma, we saw about 2/3 of the patients having unique mutations in conjunction with their shared mutations. And we were able to identify through whole exome sequencing this large group of mutations in each individual and develop fly avatars. The point was to really develop combinations that were unique and that did not rely on past history of treatments and other phase 1 and phase 2 data. We really were unbiased with regard to using multiple different drugs, including drugs that had nothing to do with cancer, but had to do with the systems that cells use to maintain and grow. Next, please. So this is an example of a patient that we did, where we identified 9 novel genetic alterations and a variant of unknown significance, and put together a multi-hit fly model avatar. We validated several of these changes on immunohistochemistry of the patient, just to be sure that we were seeing in the genetic analysis was identifiable actually within the tumor tissue. So this was all part of our validation process. And for this particular patient, we identified a dual drug combination that was effective. Next, please. Here we can see the tumor. He had had multiple treatments in the past, and we treated him with a MEK inhibitor and a biphosphonate, which, as you know, are bone stabilizing agents used in cancer and have been used often with other chemotherapy agents. We were able to put together a combination with acceptable parameters for toxicity that we thought would be tolerable. And we put the patient on this and had marked shrinkage of the tumor, as you can see in the before and 6-month scans. This response lasted for 6 to 9 months and was reported in Science Advances along with the background system analysis performed by Dr. Kagan's laboratory. So we're very pleased. We had 8 patients with colorectal carcinoma that we were able to treat with this. And while we did many more patients, we sequenced many more patients as we were developing and evolving our system. Many of these patients did not end up getting treated simply because they were late in the course of their disease when we evaluated them. But of the 8 we treated with colorectal carcinoma, we had this one good responder with this very simple combination that we developed. Next, please. In addition to the colorectal carcinoma, we did have what we called a wild card group of patients, including other diseases, and we had one salivary gland cancer patient, as well as several thyroid cancer patients that we evaluated and treated. And of the this particular patient had an adenoid cystic carcinoma. This is a rare salivary gland tumor, and these patients have a very long survival. We evaluated this thyroid cancer patients that we evaluated and treated. And of the this particular patient had an adenoid cystic carcinoma. This is a rare salivary gland tumor, and these patients have a very long survival. We evaluated this patient and identified 5 genetic alterations that we put into the fly. They tend to have a very simple genome with a reduced tumor mutational burden. And so we were able to develop a cocktail of 3 drugs. One was an HDAC inhibitor, which is used in cancer treatment. Another is an arthritis drug for which there was no evidence of anti-cancer activity in the literature. And the other was a straight up antihypertensive. And putting these drugs together in a safe regimen, we treated this patient. The 2 non-cancer drugs are oral agents. The HDAC inhibitor was intravenous. And we developed a regimen with modest toxicity. The patient had some fatigue from the antihypertensive drug and some impact on renal function and platelets, which we were able to moderate by adjusting the doses. And at the end of the day, we had a significant reduction in the patient's metabolic activity of his tumors and a stabilization at a time when he was developing rapid regrowth and growth of the cancers. He'd had no prior systemic chemotherapy for the metastases, but was requiring radiation to his bone metastases just about every month or 2 when we began treatment. And at that point, he stopped having requirements for radiation therapy. He had minimal toxicity, as I can say, and lived a full life, was working full time, going to the gym. When he began to progress at 1 year, we stopped treatment, and then he developed a very rapid progression. And unfortunately, that also occurred at the at the point of COVID involvement of New York. And so we had, I believe he passed primarily from a pneumonia, which may have been related to COVID. At that time, we didn't have good diagnostics. But we had evidence in one patient here in a salivary gland tumor of a disease that's hard to treat, for which there are really no therapies, a very good response. Not only was there volumetric stabilization of the genes, of the tumor nodules in his lung, but there was a clear metabolic inhibition of growth, reduction in metabolism as measured by PET scan in the patient, and a reduction in all of his tumor markers, for example, some of the chemical evidence of bone destruction in the blood, as well as LDH and other evidence of tumor and metabolism and tissue destruction. So he did quite well. Next, please. So I believe we are going to turn this over to Laura at this point, but I want to point out that this was very successful and that we were actually achieved a success rate higher than you would achieve by some of the other platforms. And we've evolved our technology dramatically to improve our speed of development of the regimens and also our screening process. We've now, having ended our research part of this, turned to Laura and her team to really operationalize this for patients. And they have the advantages of being able to really operationalize and optimize the technology in a way that we were not able to do, using many collaborations within our institution. So.
Okay, thank you, Marshall. And we're going to pass things on to Laura now. So if you'll just bear with us a second.
Hi, everyone. Thank you, John, and thank you, Ross and Marshall. So at My Personal Therapeutics, the cancer types that we can model today are gastrointestinal cancers, lung, medullary thyroid carcinoma, MEN2, rare genetic cancers, salivary gland cancers, and in development, glioblastoma, rhabdomyosarcoma, and triple-negative breast. So the sample requirements, we require a blood sample and also a tumor sample, the tumor biopsy. The tumor sample could be either fresh frozen or paraffin fixed, formalin fixed, paraffin embedded. And so if you'd like to order the personal discovery process for your patient, please either contact us on our email, or you can download a registration form on our website, www.mypersonaltherapeutics.com.
Wonderful. Thank you, Laura. And thank you very much to Marshall and Ross as well for their contributions. We'd like to switch things over now to questions that people have. And I'm just going to very quickly look at some of the questions that are coming up at the moment. Okay, so Marshall, the first question is to you. And this is from Greg. Basically, he's asking, at which point during the clinical process with a patient do you see the personal discovery process being used?
Well, I think in different diseases, it's going to be a different location. For example, the slowly growing tumors, I would, where you know the patient's going to have metastases and you're not necessarily moved to treat them, I would start early, particularly those where there's no established treatment plan. So for ACC or even some of the KRAS positive colorectal carcinomas, I would get the initial biopsy to work from, or the initial biopsy of the metastasis. Some of the other diseases are going to be individualized, but I see this as taking place sooner in the process. The patients, if they're relatively physically healthy and have a good performance status, are going to do better no matter what. And I would also venture to say that when one's searching for a treatment for a patient where there really is no established second line for recurrent and metastatic disease, I think this is a very reasonable option to move to as the next one. I was, we are a collaborative team. I would have treated some of the patients a little earlier than some of my GI malignancy colleagues, but I think that that is an individual decision. But I would do it sooner rather than later.
Okay, thanks, Marshall. Actually, another question for you from Andy is basically saying your studies seem to be largely with patients in late stage disease. Is there value for patients with early stage disease?
I would say yes. We had late stage disease because of the nature of our trial, which was early discovery. We had to evolve the technology, understand how to screen in the fly. So what we really did was, as I said, to go where no one had ever gone before in this process. And so we had late stage patients because that was the nature of a phase 1 trial that we were doing. For operationalization and for utilization, I think it's optimal to take the patient early in the process, make this either first line for recurrent and metastatic or second line for recurrent and metastatic. And I would venture to say that that's going to be where we'll be most successful.
Okay, thanks. A question for you, Ross, if we can. So I can see your...
Yes.
Okay. So this is from Liz. So when creating models of a patient's tumor using Drosophila, how does this compare to other model systems such as organoids and mice?
Yeah, no, that's a great question. And my laboratory also works in organoids, and we certainly considered and originally had planned to bring in organoids. What we found was, and then I'll address the other part of it, and I'll keep this brief. What we found was that the drugs and drug cocktails that we were identifying, so my lab also creates drugs from the ground up chemically, that they were acting throughout the body. All right, so we had activities when you get these cocktails on the tumor, but also on the muscle, on the liver, and then those would feed back and reduce the tumor. And when we tried those on cells in a dish, organoids and other things, they didn't look so great, but they actually looked great in flies, it looked amazing in mice and so on. So experience is, and others will back me on this, is that organoids are wonderful for understanding cancer, but maybe not so amazing for predicting therapeutic response. And I think I can show you a long list of literature on that. Now, in terms of why flies versus mice and versus other animals. So the key step in flies is to make a 12-hit model, we'll say, and then grow up 400,000 of those. To make a 12-hit mouse, I'm not aware of anybody getting anywhere close to a 12-hit mouse, but you could at least imagine it happening, sort of. But to then grow up 400,000 mice, I don't know what the cost of that would be, but I'm pretty sure it would take probably decades. So that's really not realistic. And so what happens is the people who use mice for as avatars, really you look at 2, 3, 4 different drugs, but you don't look at thousands of combinations. It's just not realistic. And so that really narrows the cocktails we've treated patients you simply couldn't find in mice. Zebrafish actually have a longer life cycle than mice, so 400,000 zebrafish, that's not going to happen. And then if you go to the other side of the spectrum, say C. elegans, assuming you have some science background, so you know these model systems, C. elegans is wonderful and they're fast. They don't have an epithelium, not a real epithelium. My apologies to my colleagues. And so you don't see any solid tumor modeling there because they just don't have a real epithelium. So there really is a sort of a narrow threading of needle, which is flies have a true epithelium and they have real organs like we do. But you can manipulate them rapidly and you can do whole gene sets. So that's why flies.
Perfect.
If I could step in here.
Sure.
There are a couple of other features of flies that are very important in contrast to mice. For example, the question may have been directed not just at growing tissues that, cell culture tumors in mice, but also PDX models. PDX is very expensive, very limited, and you can only screen a few drugs that you decided to screen. So there's no a priori drug discovery for the individual patient with PDX mice, and it's enormously expensive. The other value of the flies is as a living organism, we can do a dose response and a dose toxicity in these animals given the large number. So not only were we seeing the efficacy measured by survival of the flies, but also the toxicity in that flies that were intoxicated by the drugs, that is, who had toxic effects, literally died. And so our readout was flies alive. And finally, the third is that this can be used with robotics. They're very easy to manipulate, and so operationalizing this into a robotic process really can reduce the cost dramatically. So these were all very big positives in using flies. And finally, what I learned from Ross throughout this whole process is that the fly is really the ancestor of practically all of our biologic systems. And many of the systems that we have working in our bodies today were first evaluated and worked out in flies. And for those of you who are interested, reading Mukherjee's Gene has a whole section on developmental biology in flies and how that was the basis of genetics for decades before we began to be able to manipulate the gene. So thank you.
Thanks, Marshall. I appreciate that, and that's really helpful. I think we have time just for one more question. Ross, this is for you again. It's from Michael Castro, and Michael says some mutations are loss of function and some are gain of function with a spectrum of phenotypes. Do you model the exact mutation that is identified?
Yeah, thanks, Michael. Good question. And the answer is yes and no. So for the oncogenes, we do model the exact mutation, G12V of RAS, et cetera. So those are spot on. For loss of function, first of all, let me say most, not all, but most of those are heterozygous. We do model heterozygous mutations, which also you don't see very often. Anyway, with those, we do if our computational programs predict that it's a knockdown of function, we model it by knocking down using knockdown technology to knock down expression. And we do so, if it's heterozygous, we knock it down roughly 50%. So we really try to be faithful. If it's complete loss of function, we'll do a full knockdown. We do not model the exact mutation in tumor suppressors. So we just do a simple dial down because in that case, we can do it quickly and we can do a set of them.
Perfect. Thank you very much, Ross. And I think that really concludes today. And I'm so grateful to everyone being involved today. We want to kind of keep it short because we're obviously very aware of people's time. All the other questions that have come in, we will respond to people directly. So please keep them coming. You have our contact details, and we'll be very happy to help in any way in the future. Thank you so much for your time today.
Thank you, everyone.
Thanks, everyone.
Thank you. Thanks. Bye.